stimulating the the ipsilateral motor cortex. And if we compare the responses generated in the same muscle to stimulating on either side of the cortex, you can see that in black here, this is the response from that we would typically get from a contralateral stimulus, and you can see that the onset of that response occurs earlier than the response that to ipsilateral stimulation. And so the reason, it's been, I guess, theorized that the reason for this is that the reticulospinal tract becomes involved in transmitting responses from ipsilateral stimulation, whereas the corticospinal tract is heavily involved in delivering the early parts of that response from contralateral stimulation. So we have evidence that non-cortical spinal tract elements are involved in transmitting excitation from TMS to the muscles. We don't yet know whether these different contributors to the MEP can be independently modulated and whether that will be expressed in the MEPs that we record. And particularly, we're interested in whether differences in function can also modulate these different parts of the waveform independently. So differences, for example, between intrinsic hand muscles that are responsible for fine finger control versus more postural muscles that provide us with upright standing balance, for example. So the paper that I was asked to talk about today is this one here, where we applied some paired pulse TMS. I'm also going to sneak in another paper, if I have time, that sort of builds on this idea. The reason that we ended up collecting the data for this study, you can see Jim Stania is the final author there on the paper. So this very much stemmed from conversations with Jim. Jim had seen that so he saw some strange late arriving responses when he had people standing up versus seated, and we were trying to work out what the mechanism for that might be. And so we decided to collect some pilot data, and this is where this all originated. So this was the experiment that we ended up running. It's ended up being quite a complex experiment, so I'll try and go through it as slowly as we can and miss out as few things as I can. No guarantees. So I'll present this to you in 3 experiments. And the first one was to apply single and paired pulses of TMS to the primary motor cortex lead, the representation of the medial gastrocnemius muscle in the cortex. So we used a cone-shaped coil to deliver these responses, or these stimuli. The stimuli were very much delivered bilaterally, and we were recording from the right medial gastroc each time. Now, the way we set this experiment up was we were interested to see if we could affect early versus late parts of the MEP independently. And the way that we chose to do that in this study was to insert MEPs inside a cortical silent period. And so when we applied our paired pulse TMS, it was with that goal in mind. So to do that, so this was a conditioning test stimulus protocol. The conditioning stimulus was designed to be applied at an intensity that would induce a silent period in the target muscle of at least 100 milliseconds to give us time to insert second MEPs. And then you can see on the diagram there that we delivered stimuli at intervals of either 50 or 80 milliseconds. We also during pilot testing noticed that we did see some evidence of independent modulation, and we saw that this would seem to be dependent on the intensity that we applied TMS at. So we also ran this at 4 different intensities, all relative to motor threshold. And for the test pulses, which were delivered inside the silent period, we determined threshold inside the silent period, and then at the time of application, and then we applied stimuli either at motor threshold, which is plus 0% here, at 10%, 20%, and 30% above that threshold. So we did that at 50 and 80 millisecond ISIs. And then we also ran a range of single pulse TMS at similar intensities. So either measured via the threshold inside or without a silent period. Okay, so that was fairly complicated. Let me show you some of the results that we saw from that. So just a reminder that we were recording from the medial gastrocnemius muscle. The reason for that was that having spoken to Jim, this is a muscle that is heavily involved in standing balance regulation. And so we were interested to see that if particularly in these type of muscles, we could independently modulate. We thought this gave ourselves the best chance of doing that. And so on the left, you can see data that from one individual that we recorded when the pairs of pulses were delivered at 50 milliseconds apart, and on the right-hand side when they were delivered 80 milliseconds apart. And you can see at each interstimulus interval, you can see an evolution in terms of the amplitude of the MEPs as we increased the stimulus intensity, as you would expect. I'll just point out that in this figure here, the grey lines, I'm not sure how easy they are to distinguish, but the grey lines are MEPs that were delivered with single pulse TMS at exactly the same intensity as those delivered with a silent period. And so those are directly compared here between the grey and the black MEPs at each intensity. And so you can start to see that, even in an individual data, you can start to see changes in shape of the MEP as we increase intensity. And so what I did at the bottom here was just to take MEPs that we record, and these are average MEPs, average of 10 MEPs that we recorded at 130% of motor threshold. The grey line is without a silent period, so single pulse TMS. And then the black line is the same intensity delivered inside the silent period. These have been normalized for amplitude. These are Z-score normalized so that we can better see differences in the shape of the waveform. And you can see clear differences in the shape at both 50 and 80 millisecond ISIs. In particular, you can see that when delivered inside the silent period, the MEPs that we got heavily preferenced later portions of the MEP, anywhere from sort of 10 to 20, even out to 25 or 30 milliseconds. So I'll just take you through some of all of the group data for this experiment, and I'll try and explain this graph, and then I'll try and sort of work through this fairly quickly. So this is data collected at motor threshold, so at our lowest intensity. And what we're looking at here is the percentage change in MEP amplitude when we've compared the stimulus delivered without a silent period to that delivered within the silent period. So a positive value here represents a larger MEP or a larger amount of EMG within this period when the stimulus was delivered inside the silent period. And you can see on the right-hand side, if that scale is a bit confusing, which it certainly can be, you can see a fold change on the right-hand side, which is equivalent. And that will become more important as we move up the intensities, as we look for like how much bigger these types of responses were becoming. So you can see at motor threshold, primarily what we saw was inhibition of the response. And this is not really surprising for anyone who's worked with silent periods before. This is exactly what we expected to see when we started our pilot data collection. We expected that these MEPs would be inhibited, and we were just looking to see whether that inhibition was expressed at different times in the MEP. And so yes, we did see inhibition at motor threshold. At 50 milliseconds, that was restricted to early parts of the MEP, and you can see it stretched across the whole MEP at 80 milliseconds. So moving on now, just moving up in 10% increments, you can see a very different picture already start to emerge here. we go 10% higher than threshold, and at least a similar pattern at 80 milliseconds, largely inhibition, starting to get different responses, more variable responses in different people. But you can see that at 50 milliseconds, we're really starting to get preferential increases in the amplitude of the MEP between 10 and 30 milliseconds from the onset. So this was our first indication that, and we saw this in pilot testing as well, that this might be a technique that can independently modulate different portions of the MEP. So I'll just go up again. So when we get to 20%, you can see that pattern reinforced at 50 milliseconds ISI. All this data, I have to say, was very consistent. The changes that we saw occurred, as you could see, in essentially every person. Obviously, we had some variability across people, and that's expressed here at 80 millisecond ISIs, where you can start to see some people facilitating here, but not all people. Some people are still undergoing inhibition. And then if we move to 30% above threshold, you can see that at both ISIs now we're getting a very similar pattern of results where we get basically very little change at between about 0 and 10 milliseconds within the MEP. But we're getting very large changes now in MEP amplitude across some of the later time periods, anywhere from 10 to 30 milliseconds. The amplitude of these changes, firstly, the direction of these changes surprised us, and the amplitude of these changes also surprised us. So always our results were more obvious at a 50 millisecond interstimulus interval, and you can see that by the time we got 30% above threshold, we are getting changes in MEP amplitude in the late portion of the MEP that are 8 or 9 times larger than the control MEP. So really surprising results to us. I'll just make a quick note. You can see that there are dashed lines on these figures. And I did that because we had a couple of subjects who had interesting training backgrounds. And so one of them that I've highlighted with the black dashed line had a long history of training in slacklining. So this was someone who had really trained whichever muscles they're using in slacklining, trained in a postural sense to really try and stabilize the body on a slackline. And then the person outlined or highlighted with a grey dashed line at the bottom here is someone who'd had 10 years or more of training as a ballet dancer, and so was potentially a very different type of person with a very different neurophysiology because of that. So I just thought it was interesting to highlight that because you can see that they had very different results. And in fact, I thought at one stage that our ballet dancer was going to be the one person who didn't facilitate in the same way. But you can see that at least at 50 millisecond ISI, she did eventually facilitate in the same way as everyone else. So I guess in answer to the question, can we selectively modulate early versus late parts of the MEP? The answer seemed to be yes, so at least with using the silent period. And at this stage, and I guess still don't really understand the mechanism completely by which this occurs, but yes, this does seem to be capable of independently modulating different parts of the MEP. We were interested because part of our interest stems from the fact that TMS is so ubiquitous across the motor control literature because of the ease of stimulation of the corticospinal tract and its origin cells. We wanted to see the extent to which this applied at single pulse TMS intensities that would typically be used in a lot of studies, to see whether the same sort of modulation was evident, and perhaps that would help us to interpret these maybe in a different way. So again, I'm just on the left-hand side showing you some individual data, and at least in this individual's data, you can see some clear changes in pattern and amplitude of different parts of the MEP as we increase in intensity from motor threshold to 30% above motor threshold. The group data showed more moderate modulation. So obviously this data is a lot less clear than the paired pulse results. So, I think if you squint, there's some evidence of modulation there, but it's not clear cut. So at least in terms of single pulse TMS, it may be activating different pathways, but it would be difficult, I think, to separate that out. Okay, so let me tell you about the second part of this experiment, or experiment two. So we wanted to confirm or test out the hypothesis, I guess, that that this involved non-corticospinal brainstem mediated pathways. And one way we thought of doing that was to switch our target muscle to the FDI, so an intrinsic hand muscle, which we know receives powerful projections from the corticospinal tract and much weaker projections from the reticulospinal tract, at least. And so we repeated this experiment at one intensity this time to see whether we got the same pattern of results. Do we see at 20% above threshold the same type of independent modulation of early and late responses that we saw in the gastroc? So here are some of the results, or all of the results from that part of the experiment. I'll show you the group data first. So if you look at the bottom, you can see immediately that we didn't see the same sort of robust facilitation of late parts of the MEP that we saw in the gastrocnemius. Maybe some evidence for low-level modulation at 50 millisecond ISI, but none really at 80 milliseconds. We did see inhibition between 0 and 5 milliseconds, and you can see, I think I'll speculate, but I think the mechanism of this is relatively clear. If you look at the individual data, we saw this in most people, is that if we compare the MEP elicited without a silent period in grey to the one elicited within the silent period in black, you can see that there's a shift in the onset of a few milliseconds. That is probably due to the fact that the silent period itself has changed the membrane potential of many of the cells in the descending pathways, but particularly the motor neurons. And so the test response, therefore, the first job it has to do is to deliver excitation that restores the membrane potential back close to firing threshold and then takes it above the firing threshold. And there's a delay in doing that. So I think that accounts for a few milliseconds of delay in those MEPs, and that shows up as inhibition between 0 and 5 milliseconds. You can see apart from that, the MEPs don't really change shape very much and don't change amplitude in the same way that they did with the gastrocnemius. So for a hand muscle, we don't really see the same type of modulation as we did for the lower limb muscle. And so our final experiment was to test the idea that perhaps the facilitation that we saw in the gastrocnemius was being driven by just changes in the excitability of spinal motor neurons. So if the conditioning pulse had increased the excitability of spinal motor neurons, that would explain at least the facilitatory parts of our data. And so we looked, we didn't really expect to see facilitation because there have been other studies that have looked at H reflexes during the silent period, particularly in the upper limb, but not many in the lower limb. And so we ran this, and you can see that basically what we saw was at 50 millisecond ISI in particular, inhibition of the H reflex or suppression of the H reflex in the first 10 milliseconds, which is most of the H reflex. And more mild, but in a similar direction, some level of inhibition in most people at 80 millisecond ISI as well. No facilitation at any time period. Um, so this indicates that the motor neurons are not facilitating. They are, if anything, they're being inhibited by the conditioning stimulus, and therefore when we deliver the test stimulus, this is the background on which that's operating at a spinal level. So um, the amplitude changes that we're seeing are operating under these conditions. So if there is inhibition, actually, the facilitation we're seeing, or the increase in the MEP amplitude, um, in terms of what's happening in the nervous system, could be even greater because it's operating on that background of inhibition. Okay, so that was the whole experiment. And so then we had the question to answer in the paper of why we saw these results, and this was very difficult to answer. Um, Perhaps not obvious, and this is our best attempt so far at explaining what's going on. But if you have any questions or perhaps ideas about this, they'd be welcomely received. Uh, so just to diagrammatically indicate what we think is happening when we deliver TMS by itself. So single pulse TMS, what are we doing? Well, we know that we're preferentially activating cells within layer 2 and 3 of the cortex. If we're delivering it over the primary motor cortex, then obviously those are the cells that will be targeted, as in this study. At low levels or low intensities, the excitation generated at those superficial layers of the cortex is summated in layer 5 in our pyramidal tract neurons. And then once they're brought to their firing threshold, they deliver excitation down at least the corticospinal tract, and we think under certain circumstances, both the corticospinal tract and potentially, well, certainly maybe non-corticospinal tracts. And we're proposing that the reticulospinal tract might be involved. Fortunately, we're not the only ones suggesting that. And so this is kind of what you would get. So if you apply low intensity TMS, if you target a hand muscle like the FDI, you get an MEP. It generally looks quite simple. If you're recording differential EMG, as most people are, you get a quite simple biphasic response. That is the usual response, I would say, in the FDI. If you record from more proximal or more posturally responsive muscles like the gastrocnemius, what you tend to get is a more complex waveform, and we don't know why. But one reason for that might be that because you have these at least two pathways involved. So if you have excitation being driven along these two pathways, then potentially, particularly given the delays that exist that we know about from Stuart Baker's work in the connections, the cortico-reticular connections, then that could account for some delay in the arrival at spinal motor neurons and then muscles of cortico-reticular pathway excitation. And so we may end up with these sort of two separable or partially separable parts of the response. If we deliver high intensity TMS, then this changes the situation a little bit because we start to elicit direct activation of pyramidal tract neurons. And so if we record from epidural electrodes, then we can see we start to induce what are called D waves to a greater extent. So with direct activation of corticospinal cells. Still activating some of these upper layer cells, and the summation of all that activity again is potentially delivered along two pathways. We get larger MEPs in the hand at greater intensity, and we get larger MEPs in the postural muscle that look similar to the ones we elicit at lower intensities, with perhaps some very mild modulation, an increase of the later parts of that response. Things change quite dramatically, as we saw in our data, when we apply MEPs within the silent period. And so this is our attempt to explain how that happens. When we apply the conditioning pulse, we initiate inhibition at multiple levels of the nervous system. But certainly, one thing we do is that we suppress voluntary output from the cortex. So we're suppressing the cells in the upper layers of the cortex that are responsible for generating that voluntary activity. Uh, we are still, if we're stimulating at a high enough intensity to generate an MEP, we're obviously still generating activity in these pyramidal tract cells that is being delivered now to a hand muscle down the corticospinal tract primarily. And we think, and this is where it gets a bit speculative, we think a secondary effect of the silent period and of the conditioning stimulus in general is to suppress a tonic inhibitory pathway that exists in the cortico-reticular tract. And so what we end up with is a disinhibited reticular formation. And now our test stimulus is driving excitation through a disinhibited system, inducing some delays as per normal, and then delivering that excitation to muscles that rely on that pathway more heavily, like the medial gastrocnemius. And so what we see in this example, or at least this is what we think explains this result, is we would see a smaller or a similar size or a low level initial portion of the MEP, I guess, and then we'd see a much larger late portion of the response because of the disinhibition of the reticular formation. And then when we apply high intensity test TMS within the silent period, we get all the same effects, but we're now bypassing some of the cortical level inhibition. And so we're delivering more excitation along these pathways. And because of that, we get larger, simple MEPs in the hand. We get larger early MEPs because we're delivering more excitation down the corticospinal tract. And we get much larger late parts of the MEP because we're delivering more excitation through a disinhibited reticular formation. So that's what we think is happening. We're working on trying to understand more about this effect and whether it is the reticulospinal tract that is involved in this, and whether this explanation at all stands up to scrutiny. So, how am I going for time? Not very well.
Squeeze a bit more in.
Yeah, I'll just tell you very briefly about the second study then. So we to get at this issue, there's a lot of questions that we have still to answer about this. One of them was, if the reticular formation is involved, then we should be able to modulate late parts of the MEP preferentially if we change the postural requirements of a given task. And so this is some work done by Cassie and Nathan in our lab, and published a couple of years ago now, where we had participants in 4 different functional positions, so either seated, standing bilaterally, standing on their left leg, or standing on their right leg. And in each case, they were asked to produce the same amount of muscle activity in both medial gastrocnemiae. They were delivered feedback on a screen to help them do that. So in situations where they were standing on the left, for example, they would have posturally derived muscle activity in the left gastroc and voluntary activity in the right. And we defined sort of early and late portions of the MEP consistent with the previous study to see whether these would modulate independently. And I'll just run you through, these were some of the individual data, but I'll show you some of the group data that we got. So the main comparisons that we made were from sitting to standing, because both muscles changed the postural requirements. And then we looked at changes from standing on one leg to standing on the other leg in both directions. And so what we're looking at in this slide is the change in MEP amplitude for early and late portions of the MEP that was associated with transitioning from a voluntary contraction to a contraction where there was some postural requirement involved. And you can see that in all four of these comparisons, we saw decreases in the amplitude of the early part, the first 8 milliseconds of the MEP, and increases in the late part of the response. We saw that 4 times. And I think, like, really interestingly, I guess, and convincingly to me, we saw that in opposite directions in the right and left gastroc when they changed, depending on which muscle was involved in standing. And this is just a correlation we did. We plotted these changes on 2 axes to demonstrate. If you look at the red dashed line, that represents a line that represents changes that would be equal in the early and late parts of the MEP. Anyone above that line is changing their MEP in a way that preferences increases in the late part of the response or decreases in the early part of the response. And you can see that most of our data points fell above that line. So to wrap up, what can we take away from this data? There's obviously, when we apply MEPs within a silent period, we can strongly modulate the early and late portions of lower limb MEPs, particularly the late portion of the response is increased a great deal. If we change postural demand on a muscle, we can more moderately change the relative amplitude of early and late responses, but in a reasonably consistent way. Overall, our results do suggest that there are non-corticospinal, putatively cortico-reticular spinal contributions particularly to late portions of the MEP. Now, as I said, there's still lots of questions we have to answer here. So one possibility for that facilitation is that we're getting superimposed MEPs and stretch reflex responses. So we've just run a study where we've suppressed the stretch reflex and we've observed facilitation before and after the suppression. So it looks like that's not true. And then we still have remaining questions, like we'd love to find evidence for the mechanism by which this happens at a spinal level. Do we see, following single pulse TMS, are there 2 EPSPs that are being evoked in spinal motor neurons to postural muscles? And how are these MEPs modified, and how are changes in these MEPs modified in individuals with corticospinal tract disruption, like HSP, but also stroke, for example. And I think we're trying to work towards answering the question of whether can we use paired TMS to estimate the state of the reticulospinal system, because our current tools for doing that have issues. Some of them less direct, things like the startle response habituate very quickly. And so having an instantaneous probe of the reticulospinal system that's not subject to rapid habituation would be very useful, and particularly useful for guiding neurorehabilitation, for example. So I'll just leave you with a picture of our lab, I think celebrating one of our students publishing a paper recently, and I'll open it up to questions.
Thanks, John. Super fascinating. Really cool work. We have a bit of time for questions, about 10 to 15 minutes. So if you want to ask a question, just chuck it in the chat, and I'll read it out, or you can just raise your hand. But we have an initial question here off the bat, John, from Trevor Brown. So, and you might have just answered this, apparently. Are there any implications of this data that you can imagine will impact the way RTMS is currently used for stroke rehab? For example, what frequencies or threshold levels should be used in different situations?
That's a very good question. I haven't thought about how this applies to RTMS, actually. I mean, most of the time, repetitive TMS is being used at quite sub-threshold levels. So it may be possible, for example, to use these probes if these techniques turn out to be a reliable probe of subcortical systems, then I could imagine um, using that in conjunction with repetitive TMS to determine perhaps which pathways you've actually been able to induce plasticity in, which pathways have been involved in the changes that you're seeing. I don't know that they would particularly speak to or guide the design of repetitive TMS interventions because they're so low intensity and primarily restricted to cortical activation. Um, so it may be the case that they could help like that, but I think initially just probing which pathways are involved in the effects that might be useful for that.
It's got a question from Tim Carroll. So curious about the shape of the late response, not biphasic. Any thoughts on what it means?
Yeah, it's really interesting. If I go back actually to some of the individual data, not that individual data, there we go. Uh, well, so these ones in particular. Um, So these data, I think I presented these in a poster at some point, and someone asked if they'd been rectified, and they haven't been rectified. But as Tim points out, they're very different shape. And at least for this person, most of the negative going parts of the potential have disappeared. I don't have a good explanation for that, unfortunately. I guess just to have a stab at it off the top of my head, there may be some cancellation that we may be seeing, you know, in, I'd have to think about this very carefully, but there may be some cancellation in terms of the excitation being delivered to the motor neuron, but why that would change the waveform that we see at the muscle, I'm less sure about.
Can I ask if you played around with different electrode positions or anything like that to sort of see how that alters? Because, yeah, I mean, even in a normal MEP, you have a spread of motor neuronal discharges. So I'd probably expect a much broader spread here, but I would still think that the basic biophysics of action potential transmission along muscle fibers would hold. So, yeah, it just seems pretty odd. I mean, so it might be something really important in that. I'm not sure.
Yeah, agreed. And so that's probably something that we should play around with is electrode position. So we may, if we get the opportunity to play around with some arrays, that would be a great way to answer that.
Cool. Thanks.
Just I can't see any more questions, so I might dip in. Just to build off what Tim was saying, how does filtering matter? So filtering of the EMG signal, because even just thinking about the use of notch filters, and I know I think the general suggestion is not to use a notch filter, right? Because it distorts the waveform, it adds that annoying tail to it. But sometimes you just have to use it when there's too much noise, right, in the recording. Do you think, the use of a notch filter, but then also, what band pass settings you use can actually change the shape as well, and that could actually influence the duration of the MEP? And would that affect any of the results?
I mean, I think it definitely could. We tried to filter as lightly as possible with these data. We didn't use a notch filter, and yeah, we filtered very, very lightly. So, excuse me, I think if you filtered at lower frequencies, then yes, you would definitely spread the response out in time, and you would see obviously a smoother, but obviously a different shaped waveform. You'd lose some of the higher frequency peaks. So yeah, I think filtering does matter, and it starts to matter more when you're doing sort of these time series analysis of MEPs where you're dealing with quite um, short potentials and then trying to sort of subdivide those potentials. Then I think filtering becomes quite important. Yeah.
There's nobody else I could go again.
Yep, go ahead,
Go
Tim.
again.
Great, thank you. I really like the reticular spinal explanation for lots of reasons personally, but obviously you're talking a lot about posture. I wonder if the connections to vestibular spinal are known and if that is possible, if you've thought about that at all.
Yes, we have thought about that. So we've done. So Nathan's actually just about to submit a manuscript where we had to go at applying some GBS as well as startle to single pulse MEPs, and I think yeah, we didn't see kind of really obvious modulation, particularly with startle. We saw some evidence of some modulation, but very variable with GBS. The from what my understanding of the projections, the corticovestibular projections tend to be more indirect and weaker than the cortico-reticular tract projections. Um, so could they be involved? Yes. Um, and so yeah, so at the moment, I guess we've gone in with our explanation to kind of the strongest, maybe most likely pathway, but that doesn't rule out input from the vestibular spinal tract for sure. So it'd be interesting. I think if we ran Nathan's study again, which we may do, we'd probably probe at more interstimulus intervals so that we try and give ourselves the best chance of seeing exactly where those effects occur.
Got a question here from Janet Taylor. Have you considered that you are stimulating both sides of the head with the double cone coil, but only one side for MEPs in the hand?
Yes, I have. I haven't done too much about that, sorry, Janet, yet. But I do think that's important for generating this sort of facilitation that we're seeing. So I think there's an experiment there to be run where stimulating both hemispheres of the hand representation in particular. It would be very interesting to see whether we did see facilitation under those circumstances. We have collected some data on the upper limb, but we didn't stimulate both hemispheres in that study. So I think that's work still to be done, and I appreciate that may change the results. Yeah.
Can't see any more questions. I might just ask one last one. And if someone else wants to ask another one, let me know. But I'm trying to think about this, and this is obviously well outside the scope of the work that you've just presented. But in terms of using TMS to evoke plastic changes in different parts of the motor system, right? We always think about RTMS to induce cortical changes. We've got things like paired corticospinal motor neuronal stimulation to induce plasticity between the cortex and the motor neurons. Do you think, if we can figure out a way to obviously activate the reticular formation or the connection between the cortex and the reticular formation, does that open the possibility to induce plasticity there as well? And will that have any implications for motor rehab in stroke and spinal cord injury, etc.? Because I know Stuart Baker, whose work you presented here today, I think he pairs loud auditory clicks with TMS, and I think he's presented some data showing that seems to affect plasticity in the brainstem. Do you think your work can help lead to any more therapeutic advances when it comes to the use of TMS?
Yeah, I guess that potential is there. Yeah, so I was going to mention Stuart's work. Yeah, I forget what they call it, the clickety stim device or whatever it is. Showing that, and he's used when I presented his data. So that red part of the histogram there, as I mentioned, was put down to the effects of mechanical and auditory vibration from the coil discharge, and so he's used that and developed this vibrational technique for essentially doing paired associative stimulation of the reticular formation, and that seems to show some positive effects. So I haven't thought about it, but I guess the same principle could apply as long as you time the stimuli correctly with our type of stimulus. I will say that another reason that I feel like these later responses are brainstem derived is that and the reason that we only had 9 subjects is that this is this is quite a kind of, I always call it subjectively different experience with TMS, doing this type of, particularly at 50 millisecond ISI. It feels more like, a SHMEP, more like a brainstem mediated response where you're getting a lot of axial muscle contraction. It's not, if you had to deliver this hundreds of times, that would not be a very pleasant day. So I think the opportunities for doing that sort of thing are perhaps limited by the sort of discomfort level of the technique, but potentially you don't have to turn it up as high as we've done here either. Maybe this could be effective at lower intensities as well.
Great. I'll hit you with one last question, the final question from Tim, and then we'll wrap up. So, given task and intensity of stimulation, is there likely to be a silent period on the N on target side for the gastroc in your case? So, leaving ipsilateral projections to RF less inhibited than contralateral. Chime in, Tim, if I've butchered that reading.
I wrote it by it's non-target side, not N on target. Sorry, that was my fault.
Non-target side, sorry.
Sorry, that was a complicated question. Can you just run through that again, Tim? Sorry.
Yeah, basically, I'm just saying. So, I mean, obviously, you're measuring one side and you're showing that there's a silent period in one target muscle. I'm not sure if there's what the effective stimulation to the other side is. So, whether you think there's a difference in the level of which you're inhibiting the local cortical neurons on the two sides, so that maybe you're preferentially inhibited contralateral and leaving ipsilateral less inhibited. I'm not sure if that makes any sense or not, or whether you monitored the effective stimulus on both the sides of the body.
We didn't. So I guess the short answer is I don't know. I would expect the cortical level effect to be similar, but the downstream effect might be different because the motor neurons will be in a different state for the two different sides of the body. I guess to the extent that our explanation relies on cortical level changes in activity, that would hold for both sides. And I don't think that we would be kind of leaving one side preferentially kind of, I guess, uninhibited. But I don't like once you leave the cortex and work your way further down the neuraxis, I don't know. There would start to be some asymmetries created, and I haven't thought about it, but that may be a partial explanation for some of our results as well.
Did you pilot enough to know that you need a silent period to get this effect, or could you go slightly less? Was the intensity really important of the conditioning stimulus?
Also a good question. We didn't really test that. We're pretty much exclusively piloted conditioning stimuli that did induce a silent period. So I am not sure if lower intensity stimuli would have the same effect. It'd be interesting to try and see whether there seems to be this sort of time dependent effect as well within the silent period. So that relationship may change if we lower the conditioning stimulus intensity. But that, doing that sort of manipulation might tell us something about the mechanism that's involved as well.
Cool. So we might wrap up there. Thanks once again, John. That was a really great talk. And thanks to everyone else for joining and contributing to the discussion. I think that was quite nice there at the end. So for everyone watching, please keep your eye out on your email. We're trying to do these webinars a bit more frequently for ABSS. So that's it from me. Thanks once again, John, and hope everyone has a good rest of your day.
No, thanks a lot. Thanks a lot, Jacob. Really appreciate the opportunity to present. And thanks everyone for turning up and listening.